What is dopamine? Dopamine is often dubbed the feel-good neurotransmitter due to its key role in driving pleasure and motivation. It's tied to activities such as movement, memory, pleasure, reward and motivation, behavior, cognition, attention, sleep and arousal, as well as mood and learning. When we partake in rewarding experiences, dopamine is released, activating dopamine receptors, leading to increased neuron firing, which induces a positive, uplifting mood. The epicenter of reward and pleasure in the brain involves several key structures, most prominently in the nucleus accumbens, which is often referred to as the pleasure center of the brain. The ventral tegmental area, located in the midbrain, controls diverse behavioral events. Notably, in experiments where these areas were electrically stimulated, a hungry rat would often give up food for the sheer pleasure of self-stimulation. Sometimes you have to make your choices. It's either food or self-stimulation. The discovery of dopamine can be traced back to the 1950s, when it was discovered by a Swedish scientist, Arvid Carlsson. His work on dopamine led to him being awarded the Nobel Prize in Physiology or Medicine in the year 2000. If we're going to be talking about dopamine, it might be worth mentioning how it's made. Dopamine is made starting from phenylalanine in the body, which is an essential amino acid. Phenylalanine uses the enzyme phenylalanine hydroxylase, 2-PAH, to make tyrosine. Tyrosine is then converted through tyrosine hydroxylase, 6-PAH, which forms L-DOPA. L-DOPA is then able to undergo a decarboxylation from the enzyme DOPA decarboxylase, which results in the formation of dopamine. So if that went over your head, just to summarize briefly, dopamine is made in the body, starting from the essential amino acid phenylalanine. After phenylalanine is converted to tyrosine, tyrosine gets hydroxylated into L-DOPA, and finally L-DOPA is decarboxylated to afford dopamine. So that's the significance of dopamine. It won someone a Nobel Prize, it's made in our body from one of the essential amino acids, but how does dopamine work? When dopamine is synthesized in neurons, it gets transported into a vesicle through VMAT, which stands for vesicular monoamine transporter. If you're not sure what a vesicle is a bit like a cargo container, and the proteins that move the vesicle are like the truck carrying the containers. Once the containers get to their destination, they're unloaded, and just like the real world, containers are often reused. After dopamine leaves the presynaptic neuron in a process called exocytosis, dopamine will bind to one of the dopamine receptors, which are often referred to as D1 to D5 for convenience. The dopamine receptors are present on the postsynaptic neuron, so this is where the dopamine's going to bind. If you're interested in what the function of each of the dopamine receptors is, I'll provide a brief summary here. The D1 receptor is involved with memory, attention, impulse control, regulation of renal function, and locomotion. Everybody's doing a brand new dance now. Come on, baby, use some dopamine now. The D2 receptor is also involved with the locomotion. I don't know. The D2 receptor is also involved with locomotion, in addition to attention, sleep, memory, and learning. The D3 receptor is involved with cognition, impulse control, attention, and sleep. We also know that D4 also tends to be involved with the same processes that D3 is involved with. In the case of D5, it has a role in decision-making, cognition, attention, and renin secretion. It's probably clear by now that receptors are complex. Speaking of complex, why don't we talk about the G-protein coupled receptor complex? G-protein coupled receptors are frequently referred to as GPCRs because biologists want to save time. GPCRs are a large family of cell surface receptors involved in signal transduction. These play a crucial role in various physiological processes, and unsurprisingly, they're the target of many drugs. GPCRs can bind a wide range of ligands, including neurotransmitters. Now, what do I mean when I say ligand? In chemistry, when we're talking about ligands, normally that's something that binds to a metal center. But in the case of biology, a ligand is any molecule that binds to a receptor. So for the purposes of this video, we're talking about ligands from a biology perspective rather than a chemistry perspective. I apologize if that upsets any chemists out there. The general mechanism of G-protein coupled receptor activation involves the following steps. Step 1. Ligand binding. When a specific ligand such as dopamine or acetylcholine binds to the extracellular domain, which is just the part outside of the cell of a G-protein coupled receptor, it induces a conformational change in that receptor. The second step is G-protein activation. Once the G-protein coupled receptor undergoes a conformational change, it enables an interaction with a heterotrimeric G-protein located on the intracellular side of the membrane. This is on the inside of the cell. G-proteins are composed of 3 subunits, the alpha subunit, the beta subunit, and the gamma subunit. The third step is G-protein exchange. When the G protein coupled receptor is activated by binding to a ligand such as dopamine, a complex chain of events is set into motion. The GDP molecule bound to the alpha subunit of the G protein is replaced with GTP, leading to the dissociation of the alpha subunit from the beta and gamma subunits. The fourth step is effector activation. The GTP-bound alpha subunit and the beta-gamma subunits can now independently regulate various intracellular effector molecules, such as enzymes or ion channels. This just means that the GPCR is able to interact with other parts of the cell as a consequence of the interaction with a neurotransmitter, or other molecules. These effectors mediate the transmission of the signal initiated by the ligand binding to the G protein coupled receptor. The fifth step is signal amplification and termination. The activated alpha subunit or the beta-gamma subunits can interact with multiple effector molecules, leading to the amplification of the initial signal. The signaling cascade continues until the GTP on the alpha subunit is hydrolyzed to GDP, which is just going from the triphosphate to the diphosphate. This is where we lose energy, and I like to think about GTP as the Canadian dollar of the cell. It's not used as much as ATP, but it's still common enough. The hydrolysis of GTP to GDP causes the reassembly of the G protein complex and terminates the signal. Once the GPCR cascade is complete, several different physiological responses can occur, depending on the type of G protein as well as the cellular context. For example, let's talk about the context of dopamine signaling. Dopamine binds to dopamine receptors, which are a class of G protein coupled receptors. These receptors can be broadly classified into D1-like receptors, which includes D1 and D5 receptors, in addition to D2-like receptors, which includes D2, D3, and D4. When dopamine binds to D1-like receptors, these GPCRs couple to G-alpha subunit proteins and stimulate the production of cyclic adenosine monophosphate, although it can also be called cyclic AMP. However, when dopamine binds to D2-like receptors, these GPCRs couple to GI-alpha proteins, also known as G-alpha-I proteins, thanks biologists, and inhibit cyclic AMP production. These distinct pathways enable GPCRs to initiate intracellular signaling cascades that are specific to the receptor, ligand, and downstream effectors involved. D1 and D5 couple to G stimulatory sites. You can call this the good spot, or perhaps the G spot. And this activates adenyl cyclase. The activation of adenyl cyclase leads to the production of cyclic AMP, which results in the production of protein kinase A, also known as pKa. This is a different pKa than we talk about in chemistry, so again, I apologize, chemists. pKa production leads to further gene expression in the nucleus. D2 through D4 receptors couple to G inhibitory sites. G proteins are guanine nucleotide binding proteins, which inhibit adenyl cyclase and activate potassium channels. Dopamine also plays a critical role in neurodegenerative disorders. In the case of Parkinson's disease, when a significant amount of damage or impairment occurs in the basal ganglia, less dopamine is produced and movement disorders arise. Dopamine shortage is responsible for movement issues in patients with Parkinson's. Although the mechanism of damage isn't fully understood, Parkinson's disease also impacts noradrenaline, leading to many of the non-movement-based symptoms seen in Parkinson's patients through antagonism of the dopamine receptors. Compounds such as bromocriptine can be used to target D2, while others target enzymes involved in the synthesis or decomposition of dopamine. Furthermore, dopamine is central to addiction. Substances like cocaine and methamphetamine increase dopamine release or block its reuptake, leading to a dopamine surge in the brain's reward circuits. This can cause intense pleasure, but over time, the brain adjusts to these elevated dopamine levels, often causing tolerance. By reducing its own dopamine production, or by reducing the number of dopamine receptors, potentially leading to dependence. You might be surprised that certain foods such as cheese can also stimulate dopamine release. This is due to the protein casomorphine, which is an opioid peptide, illustrating how dopamine can be affected by what we consume. Since cheese